And okay, so it's welcome everybody. It's great to have today Leonid Mirny. And just a very short, brief biographical introduction. Leonid started his career in physics in the Moscow Engineering Physics Institute, where he got his diploma in physics, sometimes very long time ago, which was followed by a master's degree in chemistry at the Weizmann Institute of Science, where he already started thinking about applying physical concepts and theories to biological problems. After which he moved on to Harvard University, where he did his PhD with Eugene Shachnovich. And that was the group which was one of the first to start to apply very rigorous statistical mechanics to the problem of protein folding, which was one of the major problems of the day. After his PhD, Leonid moved, stayed still in Boston and was a junior fellow in the Harvard Society of Fellow as his postdoctoral position. After which he went on to a position in the physics department at MIT, where he was ever since. And he was working on a number of very interesting problems on the interface of physics and biology. And in the last decade, I guess I should say, I hope he's, okay, he's coming back. In the last decade, I should say, he was, his group and group of many collaborators, they were focused on the problem of three-dimensional spatial organization of the DNA in the human, not in the cell nucleus. And in the last 5 years, he was a co-director of the Center for 3D Structure and Physics of the Genome, joined between University of Massachusetts Medical School and MIT. And today, that's what he's going to be talking about how to use physics to understand the organization of chromosomes.
Thank you. Thank you so much, Anton. Thank you, Sid and Anton, for the invitation. It's a great pleasure talking to you. Of course, I plan to be physically in Toronto, where I have many friends, but kind of understanding the time, so we're doing this over Zoom, and sort of I'll try to kind of convey my enthusiasm about this area that I started working on relatively recently, about 8 or 9 years ago, physics of chromosomes. And so my goal for today is to tell you how exciting is the physics of chromosomes that we all carry in our bodies, and how interesting the system is. First, a quick introduction of kind of into genomics. So generally, genome is always thought as a carrier of information. There is this wonderful museum in London called Wellcome Collection, which is basically a history of medicine museum. And there is a stand where you can come and browse the genome. So the human genome is printed there in font 5, and you can come and sort of look at the information content of the genome. At the same time, genome is also a physical object. So the question is how it's organized physically in space. And sort of there is a page in Wikipedia about chromosomes. So genome is packed inside chromosomes, I'm sure you know. And again, there are kind of high school textbook is opened here that you can see. And basically every natural history museum has a stand of how DNA is packed inside chromosomes. So what do we really know aside of Wikipedia and textbooks? So we do know that there is DNA, which is about 2 nanometers in diameter. It's incredibly long. We'll talk about that. Again, this DNA is wrapped around little protein cores, and that's what forms this first level of packing called chromatin fiber. So when I will talking today about the polymer, I always mean this chromatin fiber, DNA that is wrapped around little protein cores called histones, and this provides about this gives a factor of 5 in linear compaction of this chain, because it wrapped around each histone core about 1.5 turns. So on the other end of this, we know that there are chromosomes, and the first imaging of chromosomes were drawings by Walter Fleming at the end of the 19th century. he saw chromosomes inside living cells, and they were called chromosomes because they're visible. And then a century later, we can see chromosomes at high resolution inside nuclei. So here you see in blue, you see the nucleus of a mouse cell. And inside the cell, you see two chromosomes highlighted in different colors. There are two copies of chromosome 2, a maternal and a paternal. And there are two copies of chromosome 9 in green. So you can see them. And so we see that chromosomes are somewhere in the nucleus. And the question is, how is this chromatin fiber packed inside these chromosomes? How this very long polymer is organized inside this chromosomes. What you should also realize that chromosomes do change their shape as the cell is dividing. So when the cell is doing its job, or sitting, or resting, or kind of performing some function, chromosomes are relatively decondensed. That's the picture on the left. So each chromosome occupies some chromosomal territory, like in my introductory slide, with this basically as some sort of a wire. And then when the cell is about to divide, it compacts its chromosomes into very tightly packed, elongated objects, and I'll talk more about this. Okay, so if imaging doesn't if it's a low resolution, let me know. Now the underlying, so in my talk I will focus on, kind of stepping back. So I think the physics of chromosomes is really beautiful because there's incredible long polymers that reside in our cells, and there are all sorts of interesting physical phenomena there. And historically, people thought about chromosomes as equilibrium polymer systems. And over the years, we came to realize, in the last decade, basically, we came to realize that, first of all, this is clearly very far from equilibrium polymer system. Then we found some beautiful phenomena, such as phase-separating block copolymers, and I will talk about this briefly. And then most recently, we proposed, again, together with our collaborators, that chromosomes are actively compacted by motors. There are motors that were not known before, and those that were predicted first theoretically, and these motors compact chromosomes and constantly reshape them. So it's a very complicated physical system, very far from equilibrium, nevertheless possesses certain exciting properties that can be understood using tools of polymer physics and statistical physics. So I will largely talk about these two aspects. Before going into showing you the data that we're analyzing, I want to highlight that the total lengths of chromatin. So the total lengths of DNA, so this molecule of DNA, so each cell contains about 2 meters of DNA. The diameter of the nucleus is about 10 microns. So the question is how this 2 meters are packed inside 2 microns of DNA. And another interesting thing is that so the total length of DNA in our body is really astonishing. My daughter asked me about this a few years ago, and I said, okay, so we have 2 meters of DNA in each cell, but we have about 10 to the 14 cells. This means that my DNA is like 1/10 of a light year, meaning the DNA of people on this meeting is several light years. Just to think about this, that DNA, the small molecules that contained in our long polymers, so the total length of DNA in our body is comparable to a light year. I don't know. I was shocked when I realized that. So what kind of data do we have about chromosome folding? So imaging does not really provide us with lots of high resolution pictures, but there is a technique that people say is like a chemical microscope, if you wish. But it's really a different technique. It's called chromosome conformation capture. It was pioneered by Job Decker, our colleague from University of Massachusetts Medical School. And the idea of Hi-C is relatively simple. So skipping all the biological details, I'll tell you what this method gives you at the end. It does not tell you where each piece of this polymer is located, but it tells you how frequently different parts of the genome touch other parts of the genome. So the map that you see here is the contact frequency map of in this case, of one particular chromosome, but I'll show you genome-wide maps where you can see and this map quantifies contact frequency between different regions. So regions that are frequently interacting will be darker. Those that are less frequently interacting will be lighter. So obviously we have a diagonal here because diagonal represents interactions with your neighbor. So you're very frequently interacting with your neighbor. And then the further you go from the diagonal, the less likely you are to interact with regions that are further and further apart. So these are called high C maps, and these are amazing data that tell us how chromosomes are folded. Again, it doesn't give me the whole picture of how things are in 3D, how chromosomes look like in 3D, but it gives me a very good ensemble average representation of chromosome folding or DNA folding. And it's important to realize that this is averaged over hundreds of millions of cells, each map. There are single cell maps. If you're interested, I can tell you about them at the end. So, and we again, broadly as a community, have such maps for bacteria on the left, for yeast, several species of yeast, for plant, for flies, for several cell types in mouse and human, and for a few more organisms. Again, I keep updating the slide, but I'm always a little bit behind. The take-home message from the slide is that DNA is folded in an astonishingly different ways in different organisms. And we only now start appreciating and learning this. So what I'm showing you now are the two maps. So that's a special browser for this kind of data. So on the left, you see the map, the high C map of the human genome. You see these darker squares on the left. They correspond to chromosomes. And the fact that it's darker means that every chromosome tends to interact a little bit more with itself than with other chromosomes. The beauty of this tool called high glass, developed again by Niels Gellenborg's lab in collaboration with us. The beauty of this tool is that you can have two maps loaded at once and then use the same techniques as in the Google map. You can navigate through this map of interactions, but you can also load the actual Google map. So I will be showing you this animation, assuming that it works over Zoom. So when I zoom in twice on the left, it automatically zooms twice on the right. So this way I can kind of compare features. Sorry for the low resolution, but of Zoom transmitted movies, but you should get a sense for what's happening here. Basically, you see features in these maps at all scales, at the scale of countries, states, cities, and then even regions of the city. So high C maps indeed contain information on multiple scales, and one high C map, in terms of number of pixels, equal the Google map of the Earth at 50-meter resolution. So one good experiment, so when people come to us and say, we did this experiment, we have a map for the mutant and for the control, that basically means that they are going to bring us Google Earth maps at 50-meter resolution, and so asking us to analyze and make sense of this data. So this amazing data was about 6 orders of magnitude in dynamic range, and that's what we're using to learn how chromosomes are folded. And so let me pause here for questions. So my strategy will be that I will be pausing for questions about 4 or 5 times through the talk, such that I can kind of stay connected with you, although I don't even see you. So please ask your questions, and Sid will read them for me.
Yeah, I'll be happy to read off the questions on Q&A or chat if they come up. I don't see any right now.
Okay.
So maybe we keep going.
Okay, we can proceed. Okay, so what do we do? So, okay, there is a question. Okay.
What is the criterion for characterizing interactions?
Okay, very good. So that's actually a great question. So basically what this method does, it measures proximity between two elements. And if proximity is closer than a certain cutoff distance, then it's chemically captured as an interaction. And so good experiment gives you about a billion interactions, typically between fragments that approach each other closer than, I would say, about 100 nanometers. So if two regions are closer than 100 or maybe 150 nanometers, then they have good chance of being recorded as a contact. So that's the scale, about 100 nanometers. So what we do with this data, we're building polymer models of chromosomes. So we represent this chromosome as a lone polymer. One monomer would correspond to one of such beads. Remember, I showed you this little nucleosomes, so histone wrapped, DNA wrapped around histone, so that would be one bead. Then I would impose some model-specific constraints on this. For example, I would define some rules by which different elements of the genome interact or have affinity for each other, and then I would simulate this using molecular dynamic simulations. What we've done, we have adopted a system that Vijay Panda's lab at Stanford has developed for simulation of proteins. But instead of simulating proteins, we just simulate very, very long polymers, and we use graphic cards for efficient simulations. So we can simulate polymers of up to a million monomers, and then generate a simulated high C map. So from such ensemble of conformations, I would get a map of contact frequency, and then I can compare features on this map with the features on the experimental map. If I can reproduce that, okay, that's good. So it means that the model specific constraints that I imposed make sense. And if I cannot, I can look for another model, or I can sweep through parameter space in my model and repeat this many, many times until I find conditions that allow me to reproduce the real data. And I will talk about a few examples of this approach. So that's essentially how we solve the inference problem. So instead of inferring data directly, inferring conformational ensembles directly from the data, we basically simulate this forward and then compare results to the experiment. Again, so three things. Non-equilibrium polymer systems I will not talk about today. I will talk about separating phase separation, and then I will talk about the motors. So let's talk about the phase separation. So what is the level of coarse graining? Okay, that's a great question. Typically, the level of coarse graining is 1 nucleosome. So in experiment, the highest quality data is 1 nucleosome. 1 nucleosome corresponds to about 200 bases of DNA. The total length of DNA is 3 billion bases, and 1 pixel on the highest resolution map, and 1 monomer in our simulations are 200 bases. So that's the resolution. In many cases, we coarse grain a little bit. We say, okay, maybe 1,000 bases. In some cases, we say maybe 10,000 bases. But still, even if I do 1,000 bases, a kilobase, a genome would be about 3 million such monomers. So it's going to be very hard for me to simulate the whole genome at 1 kilobase resolution. So we typically simulate short parts of the genome with some, again, so actually some of simulations I show on the next slide simulate practically the whole genome, but somewhat more coarse grained, 30,000 bases in each monomer. So the first question is about compartmentalization. So what is compartmentalization? So if I look at these maps, if you look at these maps, you would immediately see that there is a checkerboard-like pattern over here. And these checkerboard patterns are visible, and you may have noticed them when I was showing you maps before, that there is like a checkerboard patterns all over the place. And what these checkerboard patterns are reflecting is that the genome is composed of 2 types of regions. It's made of 2 types of regions. And we call these regions A and B. And these alternating A and B regions interact in a specific way, such that A interacts more with A, and B interacts more with B. So that would be this kind of, I kind of highlight these rectangles in this checkerboard. And basically what you see is that there's an enrichment of interactions between A and A, between this A and another A. And I just showed you 6 such fragments, but the whole genome can be essentially decomposed into a sequence of A's and B's. And then there is a depletion of interactions between A's and B's. And so what this means is that in space, A's stay with A's and B's stay with B's. And imaging have shown us, microscopy has shown us that A regions are typically in the center of the nucleus. Sorry, the colors are swapped between the left and right. It's my bad. A regions are typically in the center of the nucleus, and B regions are typically at the nuclear periphery. So, and that will be important. So for the next discussion. So this is called and these regions and this are called compartments. And the phenomenon of having A stuff in the center and B stuff at the periphery is reminiscent of phase separation. But it's a very special type of phase separation because it's a phase separation of polymers. So what's known about phase separation of polymers? There is a note, kind of every polymer physics textbook has a chapter on block copolymers. So block copolymer is a polymer that's made of blocks. So there is a long block of A followed by a block of B monomers and so on. And what's known about such polymers is that once you impose attraction between A and A or attraction between B and B or both, then the system would immediately microphase separate. So the difference between microphase separation and phase separation is that the sizes of phases depend on the lengths of these blocks. So if B blocks were very short, they would form little droplets within A. The longer the B becomes, they would change the shape of the microphases they form. They would become elongated. They can form a whole mesh, and then it can eventually start forming layers if the lengths of A's and B's are comparable. That's a classical picture of block copolymer microphase separation, kind of in every textbook, even in black and white, all textbooks, you would see these pictures. And this is certainly very reminiscent of what we see in the nucleus, because we see here that A, now in green, A is largely in the center, B is largely at the periphery. There are some inclusions of B at the very center. You can ignore this. This is a very special type of nuclear organelle called nucleolus. So that's kind of an exception from the rule. And now the question is, what kind of interaction, what kind of organization, what kind of physics is behind this kind of separation of A and B in the genome? And what we've shown is that this is essentially a microphase separation of a block copolymer. And in principle, the question that we had originally is about the type of interactions that drives this. So, because in principle, this can be driven by attraction between A and A, B and B, and B and the lamina. What I did not tell you is that A and B regions are functionally different from each other. A regions are typically enriched in genes. So all the reading of the genome is happening within the green part. B regions are relatively gene poor or repressed, and these regions are typically kind of moved to the periphery. And the periphery of the nucleus contains what's called lamina. It's like a matrix, and this B is glued to this matrix. So this is also known that B is kind of attached to this matrix at the periphery. I just drew it on top of a slide because it wasn't imaged in this particular experiment. But you can also assume that maybe this separation of A and B is driven by attraction of B to this matrix at the periphery. And I will spare you the details of this work because I want to talk more about the motors, but I will give you a sense for how we learn about mechanisms underlying this phase separation. And that's a collaboration again with Job Decker, Irina Solovey in Munich, Yana Fyodorova from Bulgaria, and Martin Falk, who is now who was at MIT and is now continuing as a fellow at U Chicago Physics Department. And what we've done, we looked at the structure. So first from the experimentalists, we got these beautiful images of normal cells, normal nuclei with B at the periphery. But then in many cases in biology, you can learn from some oddities. One interesting oddity is the organization of the nucleus in the retina of nocturnal mammals. So we're not nocturnal, but our cats and dogs are, and so are mice, and practically all mammals except for big apes and old world monkeys are nocturnal. So in the retina of nocturnal mammals, rods of the retina have very special nuclear organization. Their nuclei are completely inverted. So they do not have this mechanism that would attach B to the lamina, to this periphery, to this matrix at the periphery, and as a result, their B chromatin, inactive, useless chromatin, or useless DNA, junk DNA basically, ends up being in the center, and all the genes end up being at the periphery. And there was a recent paper demonstrating that this kind of organization turns rods of the retina into natural lenses. that focus light because of the different optical density of A and B regions. This is certainly another exciting physical aspect of nuclear organization, but I will not talk about that. So what we were aiming at is to understanding what leads to this phase separation just from the normal conventional picture and this rods, this naturally inverted nuclei. So again, we got high-C data for them, we got imaging data for them, and then the question was about trying to understand what's going on. So what I did not tell you is that in mouse, there is even a very special type of chromatin at the one end of the chromosome called C, or constitutive heterochromatin. And the C regions, typically, they can be labeled experimentally, and they end up being right in the center, in the very center of the nucleus, decorated by B in red, and then further another shell of A. So essentially, this inverted nuclei in rods are beautiful because we have this beautiful concentric organization of different types of the genome. And again, we get high-C data for this nuclei. And then for the model, we have now 3 types of monomers. So we have 6 types of interactions, and we would like to understand which of these interactions are the strongest and how this 6 parameters needs to be set by nature such that I could reproduce this phase separation that I see experimentally. So what we decided to do, we decided to enumerate all possible relative strengths of the 6 parameters in my model. And we did some parameter reduction along the way, so we ended up with fewer than 6 free parameters. So we fixed their values and we just swapped their relative strengths. Still, it's 720 different models to simulate with powerful computers and patient students. You can do that, and then we rank these models by their agreement to the experimental data. And what you see here is the model number, and they're ranked by the similarity to the experiment. So the first 8 models, 0 to 7, did show this concentric organization. And then I'm just showing you examples of some other models that look like they're not inverted, they're green and blue are mixed, or they're green is at the periphery, but then blue and red are mixed, so they look like mushrooms, or they look like Mickey Mouse. So you can get all sorts of architectures just by in the 720 classes of models. But the important take-home message is that among those models that agree with the data, you always see the strongest interaction between blue regions, C regions. Then the next strongest would be between B regions, and BC should be always in between. Then the next strongest would be BB, interaction between red regions. And then basic interactions between anything between green and anything else can be set to 0 without any loss of generality or changes in the result. So basically, the whole what simulations have told us is that nuclei are organized by block copolymer type microphase separation, because that I can reproduce this just by models of block copolymers. And second, this block copolymeric phase separation is driven entirely by attraction between kind of junk DNA, stuff that does not really do much, and it ends up being in the center in this inverted nuclei. But if I add lamina on the right part of this picture, if I add lamina, then in the interactions of this heterochromatin, junk DNA, with the lamina again being this matrix at the nuclear periphery, then I can nicely reproduce conventional organization. So here is a model for the conventional nucleus. Again, with green in the center, red going now at the periphery, because you tether red and blue to the periphery. You not tether, but you impose attraction between them and the periphery, and that's very much resembles what you see in the microscopy. And again, so we also quantified similarity between high-C maps. Take-home message is that chromosomes are block copolymers that show microphase separation. This phase separation is not driven by interactions between active regions of the genome. Active regions of the genome don't really attract each other, but inactive ones, B, attract each other strongly. And again, B and C together here. And lamina is important for position of the nucleus. That's a strange situation. Like without lamina, nuclei would be inverted. So inverted is essentially a default state of the nucleus. And then on top of that, for some reason that we don't really understand, in the vast majority of cells, B is tethered, is pulled to the periphery. So inverted is the default state. It's like inverted is the new conventional. And we don't know why cells are organized this way, but the underlying physics is attraction between silenced regions of the genome. And with this, I will pause for quick questions and proceed to tell you about the motors.
I guess one of the questions was, what's the significance of A and B regions?
Okay, so the significance is that A regions typically contain most of the genes, and that's parts of the genome that are being read by the cell. B regions are typically coated by special chemical marks that make sure that the cell does not read them. Why, how it works is not entirely known. Why B regions needs to be moved to the periphery is not known either. It's just at this point we just observe this phase separation. So why A and B needs to be phase separate from each other is also not known, but they are in all animal cells at least.
Great.
But at the genomic level, it's the parts that are being read and the parts that are being kind of ignored by the cell.
So another question, will you say a bit about lamina that plays an important role in this?
About what? About lamina?
About lamina?
Yeah, so basically what's happening here is that the lamina, so basically the take-home message is that lamina does not play a role in the phase separation itself. Lamina is important for inverting the inverted nucleus. So phase separation happens in inverted nuclei. Inverted nuclei do not have attachment of chromatin to the lamina. They do have lamina, and I don't want to go into different lamina types and the underlying biology. They do have lamina, but they do not have a mechanism to attach B to the lamina. And once they lose that, nuclei actually naturally convert from conventional state to the inverted one. And the lamina is important for making sure that B is at the periphery. Why B needs to be at the periphery, we have no clue. Because this inverted nuclei work just fine. They're completely functional rod cells.
Great. I think that's all we have now.
Okay, excellent. So let's proceed. So we spoke about phase separation. Now let's look at the active polymer systems. So let's zoom into this high-C maps even further. And instead of looking at the checkerboard that I mentioned, we can look at what's happening next to the diagonal. What we see next to diagonal are like little blocks or stripes or squares or triangles. It depends on how you call them. And this regions these regions are called domains. And again, in my field, they're typically called TADs, or topologically associating domains. And I don't know what's topological about them, but sort of biologists typically borrow some words from physics and mathematics to describe what they see. So I think that was one of these examples. So essentially, these are continuous regions of the genome that largely interact within themselves and interact less with their neighbors. So you can think about this as a block diagonal structure of this whole interaction matrix. And the question is, what leads to this kind of formation of the TADs? And I will not go into biology and sort of the importance of these domains. But the question is, what kind of mechanism can lead to formation of these domains? And if you think about this, there is an important problem of scales here. And the problem of scales is the following. What was known about these TADs is that they are largely established by binding of a small protein somewhere at the boundary between these two TADs. So in other words, there is a small protein here, and if this region of the genome is chopped off, then the two TADs, two domains would merge into a larger square. So somehow something present here controls whether these two regions of the genome interact or not. And this is really puzzling if you think about this from the polymer physics point of view, because there is a long fiber of the polymer, and then on this polymer there are two dots, and somehow interactions between these two monomers is controlled by a third one that is located hundreds of nanometers away. And the presence or absence of this protein determines how frequently these two others are going to interact. And this is really puzzling because it's not clear how such long interactions, such spooky action at a distance within the nucleus can work. And this, again, these proteins are called insulators. And some reviews even drew it like that. They say, okay, there is something going on here in one domain, then there is something else goes on in the other domain, and there must be a wall between them. And it's kind of funny that people say, okay, there must be some separation between them. People didn't really know how this insulation works. And we, again, decided to address this question with our simulations. So we say, okay, what if I simulate the system at some level of coarse graining, and I will impose some sort of molecular interactions and see whether that would create this kind of domains. For example, I can make boundary elements contain more polymers. I would say, okay, maybe there is some process that generates other polymers of the DNA, and that would kind of insulate neighboring regions. And it does insulate neighboring regions, but it does so incredibly weakly. So if I look a little bit, if I go a little bit away from the diagonal, I wouldn't see much of this block diagonal structure. So this kind of model didn't work. So then we looked in the model where we say, what if these regions at the main boundaries are long and flexible? And again, I simulate this and I do not get domains. So this didn't really reproduce domain structure. Then we had an idea that maybe these regions are formed by elements that just stick to each other. Maybe there are some proteins that bind here and they have affinity for each other. Would this generate domains? And the answer is no, it would not. It would generate kind of a map with dots corresponding to pairs of such boundary elements that stick to each other, but it's not going to enrich contact frequency between them and insulate consecutive regions from each other. So this kind of model didn't work either, and we continued enumerating these models, trying to come up with some mechanism that may work. And at that time, we learned about the work of John Marco, and John was interested in the question of how chromosomes are being compacted. And to explain how chromosomes are being compacted, he suggested that there might be a hypothetical process that he called loop extrusion. And the process of loop extrusion is basically an idea that there is a hypothetical enzyme that lands on DNA and forms a progressively larger loop. And then it's okay, that's a very interesting process. So let's see whether loop extrusion can explain what we're seeing inside in the Hi-C data. And then when this hypothetical enzyme falls off the DNA, this motor, then the loop opens up, and then you can, in addition to these rules, so first we just consider this system of loop extruders, and so physically there are two regimes in the system. Before talking about two regimes, there are two length scales in the system. One is the size of the loop that's going to be extruded by a motor before it falls off, that's lambda, and another is the separation, the distance between the two motors. And if lambda is much smaller than d, then they're separated by large distances, meaning that the system will be in the sparse regime. It will just form individual loops, and nothing interesting is going to happen. And then in the other regime, when lambda is much greater than d, they're going to be a dense regime, because then you're going to get loops within loops, and all the DNA will end up being inside these loops. So that's a dense regime, and we expect, in this case, DNA will be highly compacted. So this is just the underlying physics. So we said, okay, that's what can happen, but we had no idea how it's connected to reality. So then we looked at, okay, I will skip the dense regime. So we looked in the sparse regime, and in addition to this classical sparse regime, we added one more element of the model. We'll say, what if there are boundaries? And let's assume that there is a boundary, and when this enzyme comes to a boundary, it stalls at the boundary, but it continues extruding a loop on the other end. So if I were to show this with my hand, so that would be my, you can see it. So that would be so I would be a loop extruder, and I would be extruding until I come to some sort of a boundary here, but it will continue reeling DNA in on the other side. So that's the underlying model, and then we simulated that again in our large-scale simulation system, and what we found is that the system of loop extruders and borders beautifully reproduces the high CMAP. It gives this nice block diagonal structure that we see in the data. Moreover, it reproduces all these funny features that you also see in the data. So here, these are experimental data, and this is a map from simulations. It gives you the squares, it gives you dots at the corners, it gives you the stripes that you see in the data occasionally, and several other features, and quantitatively reproduces the data. So you say, oh, that's really cool. So to get these domains, what I need are loop extruders at boundaries. And so to illustrate this idea of loop extruders and boundaries, we made a little movie that I will play for you, because the new generation largely learns from YouTube. So I will play this YouTube video for you. So it plays our hypothesis, and we even say that these loop extrusion enzymes, they're ATP dependent, so they should be motors. But we even suggested which protein can serve as a motor. Say these are proteins called cohesins or condensins. And then the boundary can be established by another type of proteins shown here. Red, they're called CTTF. So that was a conceptual picture that helped us explain how these domains are formed, and we made a kind of a bold hypothesis that there should be such loop extruding motor, or that known proteins of cohesion should be loop extruders, and CTCFs are boundaries for this process, and that's why we were in 2016, and people totally didn't believe in our proposal first. But then I will tell you in the next few slides how this hypothesis was tested. But for now, I'll take a couple of questions.
I don't see any questions right now, Leonid.
Okay, given that we're running out of time, I will proceed. So what we also showed is that loop extrusion can help us solve other problems in chromosome biology. For example, compaction of chromosomes. Remember, I told you that during most of the life of the cell, the chromosomes are just random jumbles of DNA. Not random, but that's a subject of another talk. And then they get compacted into elongated chromosomes. Moreover, they form this kind of chromosomes with long scaffolds. And what we said, okay, maybe, and the question is, how can you take a random? How can you take a polymer and make it form like a sausage-like extended structure, rather than something compacted? Because if you just add an agent that kind of collapses this polymer, this collapse would minimize the surface area and would lead to formation of something that's kind of small but still spherical rather than extended. So what we did, we looked again in this dense regime and simulated loop extrusion in the dense regime. And so that's what you're going to see now on the screen. So these are simulations. On the left, you see chromosome on the left and right, you see the same chromosome in our simulations, and loop extruders are seen as this little red dots. And what you see is that initially random jumble of DNA gradually becomes, starts forming this extended structure with colors being separated, meaning that it's sequentially ordered along the chain like chromosomes are, and it forms this beautiful scaffold. So the structure, these final structures do look like chromosomes during cell division. At least morphologically, they certainly resemble the structure, and they solve the physical problem of how you can take a polymer and collapse it without minimizing the surface area. Because it's an active process that collapses it, and it reproduces this nice and extended structures of chromosomes. Moreover, when you have two chromosomes, and remember to divide, cell makes a copy of each DNA, and the question is how these two pieces of DNA can segregate. And we say, okay, if loop extrusion acts on two initially mixed red and blue chromosomes, then eventually due to loop extrusion, they will segregate. And that's what you see here in simulations. The quality of the of Zoom transmission is really low. Yeah, it's kind of updating as we speak. But what you see is that beautifully loop extrusion can separate two chromatids from each other. And you get structures that look incredibly like two separated chromatids. I will skip questions here. So then we say, okay, so there should be these proteins that perform this function of loop extrusion, and there was some indirect evidence of that from single molecule experiments. Sorry, from experiments where you can get rid of these loop extruders in the cell, so you can do high C before in the wild type. So that's what we'll show. Yeah. You can do high C before you get rid of the loop extruding of cohesion, and then after, and all these features essentially faint. All these features disappear. And if I zoom out, you'll see that I was really shocked again when I saw this data the first time. We made this prediction that should happen, but it's always pleasing to see that this is actually happening in live cells. All the structures next to diagonal, once I get rid of cohesion, all the structures vanish. That was a strong support that cohesion is a loop extruding molecule. And again, I will skip through some other experiments. And then it continued, but the people haven't really believed us because they said nobody have seen loop extrusion happening under the microscope. And it took another brave physicist, Kees Dekker from TU Delft, from Delft University of Technology, who essentially did the following experiment. So he attached DNA to the glass slide. So you see this DNA attached to the glass slide. There is a flow of buffer, and there is a protein that we predicted to be a loop extruder that's bound to this DNA, and then it sits, and then at some point ATP is added to the buffer, and then at some point you will start seeing that it leads to the formation of this extended loop. This loop is gradually enlarging. So that was a very strong, that provided very strong support for our prediction that there is loop extrusion. That was the first observation of loop extrusion under the microscope. It's not cohesion, it's condensation, it's a close relative of the molecule that we made our theory about. It was great, but there was another caveat here. Again, the theory was confirmed, that the hypothesis was supported by the single molecule experiments and by experiments in live animals, but in the theory, we assumed that the process is symmetric, that loop extrusion reels DNA in on both sides. And experiments have shown that that's largely one-sided extrusion. In other words, DNA is being reeled on only on one side. And we say, okay, that's a bummer for the theory. Can one-sided extrusion work? So we developed our theory, and that's the work of Ed Bannigan, who is a postdoc in the lab, and he will be on the job market next year, so if you guys are interested. And so what Ed did, he wrote a theory for the one-sided extrusion, and it's a beautiful kind of totally mean field, nevertheless mean field solvable model where you can get an upper bound for the maximal compaction that one-sided extrusion can do. And there is even an answer. It says that there is a number at the end of this theory. It basically says that 90% of DNA can be in the loops, but 10% of DNA will remain in the gaps between extruders. So the maximal compaction that can be achieved is tenfold compaction, approximately tenfold compaction. And simulations completely confirm this. Two-sided extrusion can give more than 1,000-fold compaction, or actual unlimited compaction, and one-sided saturates at tenfold. So basically it says loop extruders needs to be two-sided, or at least it's got to be a mixture of one-sided and two-sided. And again, so this, and it didn't take long until people did experiment on another protein called cohesin. And first, cohesin plus ATP did not form any loops until they realized that they need to add another protein to the system called NIPBL. and the simulations with cohesion and NIPBL produced nice loops. And in fact, in this case, the extrusion was completely two-sided. And they did it like on a large population of DNA attached to the glass slide. And you see that there are going to be lots of loops formed at once. This was really exciting to see this. It came out late 2019, basically exactly a year from now. It was really exciting to see this paper, this study. And since then, there were basically 6 studies that found loop extrusion in single molecule experiments. 2 observed one-sided extrusion, 2 observed two-sided, and then there was a mixture one-sided and two-sided. So it looks like the theory is now strongly supported by single molecule experiments and by experiments on cells and animals. Just to take home message, it looks like chromosomes need a motor, and this motor activity is a universal property. So these motors, these complexes of cohesion and condensation, are present in all forms of life, in bacteria, archaea, all eukaryotes. And during interphase, their goal seems to be to create these domains. And I didn't tell you anything about biology and the importance of these domains. And when the cell is about to divide, they compact chromosomes and they segregate chromosomes. The same process, it's just done by another type of loop extruders. During interphase, these are cohesins. During metaphase of cell division, these are condensins. And I hope that I convinced you that physics of chromosomes is beautiful. And there are several aspects of it, and all sorts of physics come here. Non-equilibrium, active phase separation, and our theory even inspired some musicians. So there is, if you Google for Muse IC, that's a project by Institute Curie that brought together musicians and scientists. And so there is a little musical piece wrote that is devoted to loop extrusion by a very talented composer, Amir Bitran, who is also a biophysicist now at Harvard. And with this, so if you want to listen to the music devoted to loop extrusion, Google Muse IC, Institute Curie. And my acknowledgement to all the people and our fantastic collaborators from all over the world. And thank you for your attention.